So our next section that we're going to discuss is cell size in microscopy. Although cells range in size, they are generally very, very small. And because of their small size, the biological structures can really only be viewed through microscopes, which are going to magnify those visual images. But why are cells so stinking small? And I would really like you to take the time to pause here and write your own explanation for why cells are so stinking small. But the reason why cells are so small is because they need a large surface area of plasma membrane to adequately exchange materials. Cells use their plasma membrane, that's the coating on the outside of the cell, in order to exchange materials such as nutrients and waste with their environment. Nutrients in, waste out. And cells can vary in size, but they never exceed the volume that can be nourished by the materials passing through the surface membrane. The larger the surface area, the more efficiently the cells can exchange nutrients and waste. And as the cell gets larger, its surface area to volume decreases. And so we're going to take just a moment to talk about surface area to volume. What we see here, we have two cubes of differing sizes. One has a surface area of, the surface area is calculated by the height multiplied by the width multiplied by the number of sides. In this first case, oops, sorry for that noise. In this first cell, we have a cell that is 1 cubic centimeter. 1 cubic centimeter. So because it has 6 sides, its height is 1, its width is 1, its number of sides is 6, it's going to have a surface area of 6. This other cell over here is going to be 6 cubic centimeters. So 6 cubic centimeters in length, 6 centimeters in width, and 6 centimeters in height. So 6, 6, and 6 multiplied by 6 sides. And so we are going to see that the surface area is going to be 216 centimeters squared. Now the volume, on the other hand, of this first one is going to be 1. The height times the length times the width, 1. In this case, the height times the length times the width is also going to be 216. In this smaller cell, we see a surface area to volume ratio of 6 to 1. 6 times as much surface area to volume. In this larger cube, we actually have a 1-to-1 ratio. And so the surface area to volume ratio is off. This smaller cell is going to be more efficient. Another way to think of this is to think about Reese's peanut butter cups. And this is, of course, a terrible analogy for me because I don't like peanut butter, especially not with my chocolate. I think that's just a horrible mix. But I have been told, I have it on very good authority, that those Easter eggs that come out with those egg-shaped Reese's peanut butter cups taste completely different than a normal average-sized Reese's peanut butter cup. Apparently, they're completely different. And the reason why, if you look at the wrappers, they have the exact same ingredients. They have the exact same peanut butter, the same chocolate. But what is different is the surface area to volume ratio. The larger that peanut butter egg, that thinner or that less chocolate there is to peanut butter. The smaller those Reese's peanut butter cups, the more chocolate to peanut butter that you have. So if you can think of like, I don't know, an extra large peanut butter cup, a regular size peanut butter cup, and then those itty bitty tiny peanut butter cups that you can use like for baking chips that you can use to make like peanut butter cup cookies. The smaller the Reese's peanut butter cup, the more chocolate to peanut butter you have ratio-wise. The higher the chocolate to peanut butter ratio. And that's what we care about here. What we have here, this little cell, itty-bitty cell, he is a baking Reese's peanut butter cup. He has a ton of chocolate compared to peanut butter. This guy over here, he's got a much higher peanut butter to chocolate ratio. He's got way less chocolate for way more peanut butter. So that's what we're concerned with. That's what we're talking about when we say surface area to volume ratio. How much chocolate do we have compared to peanut butter? So in order to kind of summarize this, we want the surface area to volume ratio requires that cells be very small. Large cells have decreased surface area to volume, which means decreased efficiency. The larger the cell, the bigger the peanut butter cup, the smaller the chocolate to peanut butter ratio, which is not what we want. We want lots of chocolate, very little peanut butter. And so smaller cells are going to have an increased surface area to volume ratio. They have more surface area to volume. They have an increased efficiency. Therefore, the smaller the cell, the more efficient it is. The easier it is for it to get its nutrients, the nutrients in and waste out. All right, please let me know if you have any questions about cell size and microscopy. Specifically, let me know if you are at all struggling with this concept of surface area to volume ratio, and we'll try and work it out together. All right, thank you.